Good afternoon, grade 8 students. In this presentation, we're going to talk about the different types of tests. Already we discovered the different type of tests, the different enzymes, and now we're going to determine the proper conditions for the normal functioning of enzymes and the characteristics that favor intestinal absorption. Let's talk here about the proper conditions for the function of the enzyme. So what are the conditions needed for the proper functioning of enzyme? What do enzymes need in order to function properly? Let's interpret the result found on the experiment in paragraph 1 of pages 20 and 21 to deduce the enzymatic property. Here we have two test tubes, A and B. They are placed in a water bath at temperature 37 degrees Celsius. And they contain, both of them, they contain fresh saliva. But the tube A contains cooked starch, while tube B, it contains albumin of an egg, meaning that it contains a protein. So after, for example, 1 hour, that's 7 to 8, the tube A, it gives brick red precipitate with Fehling test, meaning that it contains reducing sugar, while tube B, it gives a violet color, meaning that the protein or the albumin of the egg, it doesn't disappear, it's not digested. Why? Because here the saliva contains an enzyme which is called salivary amylase that works only on starch to convert it into reducing sugar. So here the variable factor between tubes A and B is the substrate. Here we have starch, here we have albumin of an egg. So after adding fresh saliva to cook the starch in tube A, the result is brick red precipitate with Fehling. What does this indicate? Analysis plus indication, interpret. This indicates the presence of reducing sugar obtained from the transformation of starch and the presence of saliva. While after adding saliva to albumin, here the variable factor, albumin here, and here we have cooked starch. This is the variable factor. So here we have albumin in tube B. The result is violet color with violet, meaning that the presence of proteins and or polypeptides in the albumin, and they are not transformed by the fresh saliva. So here we can deduce that every enzyme works or acts on a specific substrate. In this case, enzymes and saliva, they are salivary amylase. They can digest the starch into reducing sugar, but they cannot digest albumin. So every enzyme works on a specific substrate. That's the first property for enzymes. Second application, it's about the acidity or basicity, or it's a nutrient. It's about the pH. Pepsin, as you see here, pepsin, as we said before, it works in an acidic medium. Pepsin P, acidic like Pepsi, if you remember. So it works in an acidic medium. So here, the optimum function or the best function at pH approximately equal to 2. The speed of the reaction was maximum. While salivary amylase, as you know, as you have studied before in the handout, that the salivary amylase works best at pH equal 6.5 to 7. It's about a neutral. While the trypsin, it works best at pH 8.5, which is acidic. So here we can deduce that every enzyme needs a proper or a specific pH medium in order to work. So pepsin needs an acidic, salivary amylase needs a neutral, trypsin needs a basic. So here you have to know that for pepsin enzyme, when pH equal to 1, the speed of the reaction was minimum, was at least. As pH increases from 1 to 2, the speed increases from minimum to maximum. As pH increases from 2 to 3.8, the speed decreases back to its minimum. So that this indicates that as an increase in pH stimulates an increase in the speed of the reaction, but to a certain limit, which is pH equal to. So at pH equal to, it was maximum. For the salivary amylase, for pH equal 5, acidic medium, the speed of the reaction is the minimum. As pH increases from 5 to 6.5. 6.5 is considered around the neutral, around 7. The speed increases from minimum to maximum. And then as the pH increases from 6.5 to 9, the speed decreases back to its minimum. So this indicates that an increase in the pH stimulates an increase in the speed of the reaction, but to a certain limit, about 6.5. So it works best at 6.5. About 6.5, it goes back to its minimum. So let's talk about the variable factor about pH for trypsin enzyme. For trypsin enzyme, for pH equals 6, the speed of the reaction is minimum. As pH increases from 6 to 8.5, the speed increases from minimum to maximum. As pH increases from 8.5 to approximately 10, the speed decreases back to its minimum. So what does this indicate? This indicates that increase in the pH stimulates increase in the speed of the reaction up to a certain limit, is 8.5. So what is the enzymatic property deduced here? That every enzyme needs a proper pH medium in order to act. So every enzyme needs a specific pH medium, and every enzyme till now needs a specific substrate. So here the variable is the pH. Moving to the activity of the enzyme versus temperature. The activity of the enzyme versus temperature. Here the activity of the enzyme was maximum at temperature 37 degrees Celsius. Here at 37 it was maximum, and then it decreases to its minimum at 60, and it was minimum at 0. So here, as temperature increases from 0 to 37, the activity increases from 0 to 100 degrees. And as temperature increases from 37 to 60, this activity decreases back to 0. So what does this indicate? This indicates that increase in temperature stimulates an increase in activity of the enzyme up to a certain limit, which is 37. So every enzyme here needs a proper temperature in order to act, needs a specific temperature in order to act. So here we have 3 properties for enzyme. First one, every enzyme needs a specific substrate. Every enzyme works on a specific substrate. Every enzyme needs a proper pH medium, whether acidic, basic, or neutral. 3, every enzyme needs a proper temperature. So you have to keep in mind that the optimum temperature in this case study where we study here, the optimum temperature is 37, since the enzymatic activity is at its best condition. Inactive is 0, since the enzyme is inactive at temperature 0. And 60 degree is called denaturation or destruction. You have to know both terms. So at high temperature, the enzyme is denatured or it's destroyed. Best or optimum temperature, the best temperature is 37. Inactive temperature is 0 because the enzyme is inactive at 0. And 60 degree is called denaturation or destruction temperature, since the enzymes are destroyed at high temperature. So you have to know that if the enzyme is inactive at cold temperature, say is 0 or -1 or -2, it can work again if it's replaced at normal temperature. For example, I put an enzyme at temperature 0 degree, okay? And then I place it back at 37. The enzyme can work again. So it's normal. But if the enzyme is placed at very high temperature, the enzyme is destroyed or denatured. So it cannot function anymore because the high temperature destroyed the enzyme. The enzyme is not functional anymore. You have to know this. Moving to chapter 1, activity 4, from food to nutrients. It's easier for the food to be digested into smaller pieces to be digested. So it's easier for the juices to help them to be digested into smaller pieces. We have 2 types, either mechanical or chemical. You take this in grade 7. So mechanical digestion breaks down food into smaller pieces, and it will keep the food moving along the digestive tube, mouth, esophagus, stomach, small intestine, large intestine, and then to the anus. While the mechanical digestion involved in the mouth, you have something called chewing. In the stomach, you have something called churning. In the small intestine, we have something called peristaltic movement. Chemical digestion. It involves the digestion of food by digestive juices. It breaks down food into simple nutrients. It occurs in the mouth, the stomach, and in the small intestine. How are these two types of digestion needed? Mechanical digestion, you have to know this. Mechanical digestion of food, so the digestion of food into smaller pieces will facilitate, will accelerate the chemical digestion and allow the digestion to finish faster. You have to know this idea. So mechanical digestion of food facilitates the chemical digestion and allow the digestion to finish faster. Moving to activity 5, the route of nutrients. You have to know that we have 4 characteristics that favor the intestinal absorption, the absorption of nutrients at the level of the intestine. The small intestine, they have thin wall of intestinal cells. They have large surface for exchange. They are highly vascularized, meaning that they are rich in blood vessels. We have to study these notes, the characteristics. Highly vascularized, meaning that they are rich in blood vessels and lymphatic vessels. And the 4th one, which is moisture. Okay, so here, the routes of absorption, either through the blood or through the lymph. Blood absorbs all the nutrients except fatty acid and glycerol, while the lymph absorbs fatty acids and glycerol only. So here, as a reminder, you have to know that characteristics that favor intestinal absorption: thin wall of intestinal cell, large surface for exchange, highly vascularized, and the moisture. You have to know that mechanical digestion facilitates the chemical digestion and allow the digestion to finish faster. You have to know that chemical digestion is the digestion using digestive juices. And you have to know that mechanical digestion that breaks down food into smaller pieces. And we have mechanical digestion in the mouth for chewing, churning in the stomach, and peristaltic movement in the small intestine. And you have to know that the high temperature destroys the enzyme. It's not functioning again, even though when it's back at 37, while inactive temperature, let's say 0 degrees, it can work again if it's placed at 37. You have to know that the enzymatic properties, we have 3 enzymatic properties. Every enzyme works on specific substrate. Every enzyme works on a specific pH medium, and every enzyme works on specific temperature. Okay, guys, so these are the notes needed for this session. You have to know this session, and you have to revise all the notes for digestion since they are needed for our quiz.